da vida, né, sobre os diferentes aspectos da filogenia dos grandes grupos. E agora de manhã nós teremos conosco o Dr. Sven Gould. O Dr. Sven Gould é um grande pesquisador de temas relacionados à endossimbiose primária e secundária, né? Também diferentes tipos, relações aí entre endossimbiose, por exemplo, com lesmas do mar, né? Então, trabalha muito com a questão de membranas, diferentes aspectos de origem, são temas extremamente interessantes. Dr. Sven Gould, ele é graduado em ciências biológicas, ele é biólogo também, né? Pela Philips Universität Marburg, e lá na Alemanha. E também fez o doutorado, o PhD dele, pela mesma instituição, trabalhando com Protein Target. O que acontece? Só uma curiosidade, a Universidade de Marburg, ela foi fundada em 1527, então ela está entre as universidades mais antigas do mundo. Bastante interessante isso também. E o Dr. Sven Gould, atualmente, ele trabalha na Heinrich Heine Universität Düsseldorf, lá no Institut for Molekulare Evolution, né? Na verdade, em alemão. Então, com vocês, eu vou trazer agora o Dr. Sven Gould. Welcome, Dr. Sven. Thank you for accepting our invitation for our course, and welcome also to Brazil and Latin America, even virtually. Your microphone is muted.
Yeah, good. I said good morning, I guess, looking at your timeline.
Yeah, we are 9 a.m. o'clock, and there in German is 2 p.m. o'clock now. Then, welcome, and we have the words to make your explanation. Thank you.
Good. Yeah. Hi, everybody. It's a pleasure to be invited. I already mentioned earlier that it's actually one of the favorite parts of my job to contribute to the bigger audience, the science that we do on an everyday basis in our group. So, as you will see, or as you know, the program was announced with primary and secondary endosymbiosis. I will expand a bit on that. Yeah. Simply because, to give you some background, what we do is evolutionary cell biology. So I'm a cell biologist by training, but always, basically ever since I started my PhD, became interested in evolutionary aspects. So all of the cell biology that I do, or that we do in the group, is embedded in an evolutionary context. So we do basic cell biology on several protists. We work on the terrestrialization aspects of algae, so the transition from water to land in the algae and plant world. We do genomics on different levels, but we also sometimes plot evolutionary scenarios, as here in the bottom paper where we were discussing the origin of the eukaryotic cell. So it's a pretty broad spectrum that our work covers. So we never really, for instance, focus on a single specific organism, but we pick up a topic and then try to identify the best organism to answer these evolutionary questions for us. So today, it's a bit of a challenge for both sides, but I thought it's this is a good opportunity to give you a broad overview of the stuff that we do. I won't be talking too much actually about the exact science that we do, so I won't be presenting papers because you can read them up all online. I'm trying to just give you a broad overview of 4 billion years of evolution. So, because if you want to understand the impact of endosymbiosis on this planet, it's good to have a broader picture. Yeah, so we'll be walking through these 4 billion years of evolution. So starting with the origin of life for a few slides, and then we'll move over to LICA, which is the last eukaryote common ancestor. So this is eukaryogenesis. This is where we have the first endosymbiotic event that really left an imprint in evolution. And then we will move up to the origin of plastids, and then at the end of the day, secondary endosymbiosis. I'll be very briefly, or maybe 2 or 3 slides, be talking about land plants. Yeah, so the shift from water to land. So 3 main sections, and every time that this picture pops up, you know basically that we're moving into the next section. Yeah, as I mentioned, origin of life and eukaryogenesis to begin with, then primary plastid endosymbiosis, and at the very end, or not the very end, but then I'll be moving on to secondary plastid endosymbiosis, and at the very end, then, if the time allows, we'll have a look at some peculiar cases, what happened actually with some of these critters. So to begin with, the origin of life. I mean, this is one of the aspects I think that is just deeply entrenched into the human brain, and that is the question of where we come from. Yeah, I mean, if you think about it, every cultural background has its own religion, and religion is always at the core wanting to know where we come from and where we are going. And in biology, this is nothing, there's no big difference. Yeah, so investigating abiogenesis, the origin of life, is basically this religious aspect, wanting to try to understand how it all began. For today, yeah, we can simply plot it this way, that the origin of life to a degree also translates into the origin of the prokaryotic cell. Because only then did we have a free living form that evolution could select upon. Yeah. But very early on, yeah, we had at the bottom of the picture here, what you can see is basically where we get this shift from a geochemical background, yeah, so where geochemistry evolved into biological chemistry. Yeah, this is the very origin of life. And from what we actually understand today, at the very early beginnings, this was independent of the plasma membrane, yeah, because it looks like it was happening in some kind of rocks, yeah, so the smokers at the bottom of the oceans, where you have little caveats, which actually, when you cut these rocks, they roughly have the size of prokaryotic cells, so a few micrometers in diameter. So perfect little reaction chambers, if you want. But early on, it looks like that then this first life, which wasn't free living, separated itself into 2 lineages. Yeah, so where we get here in blue the bacteria and in red the archaea. Yeah, we know this, that we had this early divergence into these 2 lineages simply because the plasma membranes of these 2 are very different in nature, yeah, in terms of their biochemical nature. And also the enzymes that synthesize these lipids, they're deeply divided. Yeah, so it's a deep dichotomy between these 2 lineages. But the origin of life was only there once, and this is of course evident from, for instance, the ribosomes or the genetic code, yeah, that both of these use. But these 2 will come or will be important later. But until here, we have no endosymbiosis. Yeah. If we then move on to the first kind of trees in biology, here pictured Darwin, that you all probably know. He was actually one of the first ones to really, he wasn't the first to do trees, yeah, because of course family trees, they go back way earlier. But Darwin was the first to have this tree thinking, or took the tree thinking into the biological world. Yeah? And he has this famous sketch that he made. It's in one of the notebooks that he had on his journey to the Galapagos Islands, actually. And one thing that many are not aware of, simply because it's, you know, not the everyday that you, or not the usual way that you think about Darwin and his works, is that most of his work was actually based on vertebrate evolution. Yeah, so that's what he observed. He was a great observer of nature, and that's also what he plotted his models on. And again, yeah, in these, there was no endosymbiosis playing a role. At that time, no one had any idea that endosymbiosis could be evolved into anything, because it took another 50 years for that to emerge. So here, what you see on the right-hand side, this is the only picture that is actually in his book, this famous book on the origin of species, which really was groundbreaking and defined the evolutionary theory that we still work with today from 1859. But all of these lines that you see, they're entirely based on vertical inheritance. Yeah, so mother, father, children, and so on and so on, and next generation, next generation, next generation. Pure vertical inheritance. And as we will see later, endosymbiosis is the very opposite of vertical inheritance. Then, 7 years later, here on the bottom right is also an iconic picture by Haeckel from 1966 that he drew. And he was the first one to really visualize the broader picture of evolution. Yeah, so where he came up with these big three branches in the tree of life and really plotting a whole tree of life entirely from everything that was known at that time. Plantae, protists, and animals. And then here at the bottom at the root, as you can see, he didn't really bother too much about prokaryotes because also not that much was known about prokaryotes. If you ever find your way to Jena, which is in the eastern part of Germany, I can highly recommend to you. His house is still standing, it's part of the university, and his office is basically there. It's part of the museum or part of the university if you want, that you can visit. Yeah, so highly recommend it. This desk is where he drew the picture, and as you probably know, he was also a fantastic artist and painted many famous pictures on which you can buy books. But again, in this tree of life that he pictured, there was again no endosymbiosis involved. The first time that endosymbiosis came into play in terms of evolutionary biology and plotting it as a picture was in a famous paper by Konstantin Merechowski in 1905. At that time, most of the scientific literature was actually in German because at that time we hadn't yet started 2 world wars and hadn't lost them. So his paper was on the nature and origin of chromatophores in the plant kingdom. Yeah, so as you can probably tell, he was thinking about the origin of plastids at that time. And he was the first one to include endosymbiosis and to think about that maybe the organelles that we see, these green chromatophores in plants, that they could be once free-living cyanobacteria. So if you look at the picture here on the right, this is exactly what you see. So you've got 2 trees. Yeah, we don't need to worry about the details for today. On the left, a bit of tree, and on the right, a bit of tree. And here you see these lines. Yeah, so the bottom one says first symbiosis, and the other one says zweite Symbiose, or second symbiosis. And it was visionary for his time. It really is, if you think about it, what else was known at the time to come up with this model, which in the broader scheme has been proven to be correct only, 60 or 70 years later. Yeah. So the first symbiosis, where he didn't really know exactly what it was, but he knew that something must have happened. This is actually the origin of the eukaryotic cell. Yeah. So he knew that there's this prokaryotic world and something must have happened. He was plotting the first symbiosis to get, in his case, to amoeba. And then he visioned that amoeba phagocytosed a cyanobacterium, which gave us the rise of the plastids. Yeah, so really a big paper for that time. It is still, if you read it today, there's an English translation available online for free. Highly recommend it. It's up to date. It really is. It was long forgotten. Yeah, so from 1905 until to the 70s, roughly. Much had to do, actually, there were historic reasons why it was suppressed. But biology at that time just couldn't imagine, that this could be true. So it took until to the 70s when Lynn Margulis came up to write it all up in a new paper where she was thinking about the origin of eukaryotic cells, that it then became more a common research topic again in evolution. So to get back to the tree world, the next big step was then actually by the introduction of the 3D tree. Yeah, so that is where Wöse and his colleagues they plotted based on just 16S RNA sequences and 18S RNA sequences. This famous tree where they saw that there was a deep divide between the bacteria, the archaea, and the eukaryotes. Yeah. And this is the 3D tree, and it's a discussion that is ongoing. You'll see that on the next slide. Simply because we have one root, but then we have 3 independent domains. Basically, what you can see here, and this is what the 16S data at that time was telling us, or 18S as well, is that there was one branch from which the eukaryotes emerged, and then the archaea came out of it. Yeah, so these famous 3D domains. Sorry. And just a few years ago, 2015, 2016, a new group of archaea, yeah, so if we go back for a second, so here we have all of the archaea, in red again, a new group of archaea was discovered, the so-called Asgard archaea. Yeah, they're called Asgard archaea because names, they are Loki archaea, Thor archaea, and so on and so on. So all from the Asgard kingdom, yeah, if you know the movie as well, the Hollywood movies. And they changed the whole picture a bit because now we suddenly had a 2D tree. Yeah, and you can see that here in the right, this little brown triangle, this is actually the eukaryotes. And you can see that the eukaryotes here are sitting within the archaea. Yeah, so going back once again, here you have the big archaeal branch and the big eukaryotic branch and the archaea emerging out of it. Yeah, on what the Asgard archaea then showed is that the eukaryotes actually branch here within the archaea. So that is why we now also, the majority of data today tells us that it's a 2D tree. Yeah. What was also very special about the Asgard archaea is that they were found to encode several proteins which until then were thought to be uniquely encoded by eukaryotes. So you have 6,000 gene families roughly, which you only find in eukaryotes and not in prokaryotes. And these archaea changed a bit because some proteins were identified that were suddenly encoded also by prokaryotic lineage, these Asgard archaea, which also branched together with the eukaryotic group. And the interesting thing about these proteins is that many of them are associated with fundamental cell biological processes or membrane manipulations, which were thought to be unique to eukaryotes. But at the end of the day, the consensus of the last years really has been reached, and there is no reason to doubt this anymore, that eukaryotes, and that takes us slowly to the topic of today, that eukaryotes are the result of endosymbiosis. Yeah? And here pictured nicely by Mona, a postdoc from our group, you have the bacterial kingdom, the archaeal kingdom, and they just came together and became one to provide us with the foundation from which eukaryotes could evolve. And this is an actively discussed and worked on topic, of course. Why is that the case? Because I think Nick Lane, in his book, he formulated it the best way. He called it the black hole at the heart of biology. Yeah, and that is trying to understand how the prokaryotic life transitioned to eukaryotic life. We now understand that LICA, as I just explained to you, is the product of endosymbiosis, yeah, the last eukaryotic common ancestor. So we know that the merging of the proteobacterium into an archaeal cell gave us the origin of eukaryotes, but how exactly this transition worked is not yet known. And it is so fascinating if you think about the very first slide that I showed you in terms of the timeline. For 2 billion years, we had prokaryotic evolution. And I'm pretty sure that if we would have a time machine and we would hop on in a time machine and travel back, let's say 3.5 billion years ago, that the prokaryotes that we would sample would pretty much mimic those that you would sample at any pond today. Yeah, so it's hard to explain why they had so much evolutionary time at their disposal and they didn't use it before endosymbiosis occurred. And then once we had endosymbiosis happen, we get this whole new radiation, yeah, this explosion of diversity among eukaryotes, the many different eukaryotic supergroups, and then also here, top right, the pictures, just all of this macroscopic complexity that you just do not see even remotely among the prokaryotes. And that is the question how that really happened. Now, if you focus on the left first, yeah, I know it's all very small, but the idea is also not that you see every detail, but just imagine that each line at the end of the day represents one single model for the origin of how eukaryotes evolved. So from prokaryotes to eukaryotes, what happened. We now know that many of these models cannot be true any longer, but at the end of the day, what's interesting about them is they basically boil down to two different possibilities. One is a phagotrophic origin of the mitochondria, yeah, so how we got to the endosymbiotic origin of mitochondria, and the other one is the syntrophic model. Now, what does this mean? phagotrophic origin is basically at the very bottom. Early on, you had an archaeal lineage that autonomously, somehow, stepwise, and gradually evolved complexity to a degree that it allowed these cells to phagocytose. And only then, yeah, so you can see here that we still have an archaeal cell, but it already has a nucleus and some Golgi apparatus, etc. So this then was able to phagocytose prokaryotic prey, and one of them established itself as the mitochondria, yeah, and then from there on, eukaryotic evolution as we know it. On the right-hand side, this is the other scenario. At the bottom, you have, imagine a biofilm, yeah, which is the norm in the prokaryotic world. Yeah, no prokaryote lives by itself. So you have a biofilm, you have a syntrophic interaction. Yeah, so two partners that profit from each other by exchanging metabolites, for instance. And then one of them gets integrated into the other for reasons we cannot get into today, and how that was maybe possible, but we do know that prokaryotes have this ability. And then the big difference to the left model is that actually the origin of this endosymbiont, yeah, so the endosymbiotic entry of what we now know is the mitochondrion, then allowed and triggered and fixed all of these compartments that we see in eukaryotes. And at the very end, you get to your picture book protist that we now know today. Yeah? So this is these two models. I don't like the term that they compete with each other, but it's basically, I think if you want to understand the topic, it's the easiest way how to digest it. You either have an autonomous origin, mitochondria coming in very late, or you have a syntrophic origin, and then the fusion actually made the eukaryotics possible. I personally, based on the data that I see, think that mitochondria early models are just the best in terms of providing a full service theory. Yeah? To me, it's the easiest how we can explain these transition events, and what they take into account is, for instance, the singularity of eukaryogenesis. Yeah? If prokaryotes could evolve it by themselves for some reason, why did it only happen once? Yeah? It also explains why we have no intermediates among the prokaryotes. We have hundreds of eukaryotes in culture, none of them even remotely look like the eukaryotes that we know today. Even the Ascaricia that are now in culture, they are all really prokaryotic in nature. The universal dispersion of mitochondria across eukaryotic diversity. There is no eukaryotic lineage that lacks a mitochondrion. Yeah? So we now know that and understand Lika had the mitochondrion, so it's deeply embedded into the origin of eukaryotes. Many aspects of eukaryotic cell biology can be explained by the presence of a mitochondrion. That will be on the next slide. And the Ascaricia that we now have that were identified, despite the fact that they have these ESPs, which stands for eukaryotic signature proteins, so the proteins that I mentioned, which were until then only found in eukaryotes, are now also present in prokaryotes. What they confirm is it's a syntrophic scenario, because the cultures that were isolated, you can only grow them basically like what you see here on the bottom right, so in a syntrophic association, so a bacterium and an archaeon. And what these pictures also showed is that they lack any cellular internal complexity, so they do not have any endomembrane system, which is such an iconic feature of the eukaryotic cell. And getting back to the many aspects of eukaryotic biology, why can they be explained by a mitochondrion? Because if you think about it, if you want to house an endosymbiont within your cell, you have to provide it with several solutions. Yeah, it was a free-living prokaryote that, for instance, needs to be fed, etc. Yeah, it was a free-living prokaryote that was secreting vesicles into the environment. Every single prokaryote that we know secretes vesicles as a part, for instance, of waste disposal. So vesicle secretion that we observe in eukaryotic cells is the recycling of mitochondria-derived vesicles and serves lipid exchange. The peroxisomes that we see in eukaryotic cells, they helped to compartmentalize beta oxidation, which is problematic biochemistry, which damages the mitochondrial DNA. So it was basically physically removed from the mitochondrion through peroxisomes. The autophagosomes that we have in eukaryotic cells, they are there to recycle damaged mitochondria, because of course every now and then some mitochondria in your cells break and you have to remove them. For that you need an autophagosomal machinery. The nucleus, separation of transcription and translation, and so on and so on. I won't go through them in detail. The bottom line is that all of these eukaryotic traits that you observe today, which define the eukaryotes, are probably really just an evolutionary solution to housing an endosymbiont. And I'll get back to this. I'll get back to this later when we talk about the origin of plastids. Yeah, but if you look at eukaryotic biology, it really is not only defined by mitochondria, but also very likely was made by mitochondria. So. And that's what I meant by early on when I said, thinking about the evolutionary timeline, and if you want to understand what impact endosymbiosis had on this planet, 2 billion years of prokaryotic evolution, and if you were looking at Earth from space, for instance, you wouldn't have seen much. It would just have been some rocky surface, and you wouldn't have spotted any prokaryotes, of course. But the first endosymbiotic event, which really established itself, provided us with eukaryotes, and with that provided us with a complex form of life, the eukaryotic branch, which is characterized by the compartments that I just mentioned. And the beauty about the eukaryotic cell is that it is really an amazing mix of bacterial and archaeal biology. It's here shown, so anything that is in red is again archaeal, anything that is in blue would be bacterial. But that's too much to get into detail for today. But it really is amazing thinking about it. Origin of life meant you had these two lineages emerge, bacteria and archaea, and then 2 billion years later they fused again to come up with an entire new branch, which gave us the eukaryotes. And with that, we would move on to the next part. So we just had eukaryogenesis, a very brief overview of what eukaryogenesis means, but the fusion of two prokaryotes into a eukaryotic cell and its big impact. And now, primary plastid endosymbiosis. Let's move on to that before we look at secondary plastid endosymbiosis. Eukaryotes became phototrophic, not again by inventing their own biochemistry, but as eukaryotes often do, but by stealing it from the prokaryotic tree of life. And in this case, they simply did it by engulfing an entire cyanobacterium and slowly transforming it from a free-living cyanobacterium into an enslaved plastid, the way that we know it today. This occurred some 1.2 to 2 billion years ago. It's really difficult to pinpoint. The fossil records speak different stories. I think it's fair to say it was 1.8 billion years ago, from what I can tell. But it's enough to understand that it's, again, an endosymbiotic event that is really old and also pretty close, actually, to the root of eukaryotes, which is also why the branch of Archaea plastida, so all of these combined is what you would call the Archaeplastida, is one eukaryotic group by itself. I can actually quickly, if we go back here. So here on the right-hand side, this is what was already pictured. The plastid came in here from the cyanoactrial branch and gave us the Archaeplastida. And we don't know the exact rooting of the tree here. You can spend years trying to understand it and you still don't. And you can also discuss it for years and you will still not have one opinion. But nevertheless, the origin of the plastid is very close to the origin of eukaryotes itself. We have 3 primary lineages that survived since the original establishing of the cyanobacteria. They are the glaucophytes, chloroplastida, and rhodophytes. I would bet, if we would be in one room and I could ask you this live, that the minority of you would have ever heard of glaucophytes. There's only a handful of labs on the planet that actually work on them. They are a bit like the dinosaurs, if you want, that have survived for a long time. Their plastids actually really still look a bit like free-living cyanobacteria, and I think only roughly a dozen species or so have been described of glaucophytes. So they are ubiquitous, you find them all over the planet, but still just a few species, and they get along just fine the way that they are, probably. Rhodophytes is a different story. They are actually pretty important in the ecological aspect, and we'll see that they will be very important when we move on to secondary endosymbiosis. And an interesting branch in terms of their biochemistry, and they do many things differently from the main lineages that most of you will know, the Chloroplastida. You might not know them as Chloroplastida, you might know them as the green lineage or whatsoever, but you surely also all know land plants, and they actually at the end of the day emerged from green algae and belong to the Chloroplastida. And what I just mentioned in terms of eukaryogenesis, and one of the reasons why I wanted to talk about it early on is that all of the compartments and all of the eukaryotic traits that I just explained, they were there and probably evolved as an answer to the first endosymbiont, which gave us the animal cell here on the right. But when the plastid came in, not much had to change, because everything that you needed to serve a new endosymbiont, the plastid in this case, the chloroplast, was already there because the mitochondrion was, of course, first. So the switch to eukaryotes was a completely different one, comparing from going then to a eukaryotic cell to a plant cell. We also know that many things like dynamin-based scission, thyroredoxin-related redox regulation, etc., that plastids use were copy-pasted, so to speak, from mitochondria. So lots of the things that the cell needs to house the mitochondria were then used to also house the plastid. So this also speaks towards the fact that many of the eukaryotic compartments are an answer to endosymbiosis, and why you just do not see them in the prokaryotic world, because you only need them when you have an endosymbiont. One question that every now and then pops up in the literature is, it's just an evolutionary biologist's curiosity, is what is actually the closest living relative to the cyanobacterium that was engulfed so long ago? And some questions that we try to answer is, was it really CO2 fixation that drove the endosymbiotic fixation, or was it maybe nitrogen? Because we all know that nitrogen deprivation is a problem in plants, in particular because of photosynthesis. And so these are just competing questions, and why I want to know which free-living cyanobacterium is the closest sister lineage to the one that was engulfed. Or another question is, where did this actually happen? Did it happen in a freshwater or in a saltwater environment? And why is it so difficult? Any one of you that has done a phylogenetic tree would say, oh, we just take the sequences from cyanobacteria and today's plants, and then we just plot it, and we should see exactly where the cyanobacterium comes from. Well, one thing that you need to keep in mind when you're trying to analyze old evolutionary processes, so very old in this case, like in the origin of plastids, is the problem of mosaic genomes. So that's a term that you'll find and might want to remember. Phylogenomics on ancient endosymbiosis always means that you have the problem of encountering mosaic genomes. Now, what does mosaic genomes mean? Well, horizontal gene transfer is what prokaryotes do on a daily basis. It's the way how they recombine. It's their way of escaping Miller's rigid. Eukaryotes, such as us, we have 6. Prokaryotes don't have this. They have horizontal gene transfer, or sometimes in the literature you also find lateral gene transfer. It's the exact same thing. And here on the right, I just put a picture of the different ways how prokaryotes transfer DNA from one cell to another. You have transformation, conjugation, and so on, but that would be part of a microbiology class. The point being is, it's common biology in prokaryotes. If you're thinking about multi-resistant strains, the reason that they're multi-resistant is because they can just exchange DNA and profit from the best DNA available to them. So that is part of prokaryotic biology, and it always probably was part of prokaryotic biology. There's no reason to think why it should have evolved later. It's very likely that with the origin of prokaryotes, also horizontal gene transfer emerged. So if you now think about that, you want to plot an event that occurred 1.8 billion years ago, keep in mind that at that time, your endosymbiont, basically, you took a picture shot of a cyanobacterial genome, And all of the free living relatives, so all of the sister lineages that you can culture today, yeah, you go out to the sea, to the pond, to a puddle, and you isolate the cyanobacteria and you sequence them, they all have been going through horizontal gene transfer events for 1.8 billion years since they diverged from the cyanobacterial lineage, which gave us the plastid. Yeah, so it's a wild mix of DNA. You constantly chop it up, rearrange, and pass it on. This is also why we have pangenomes among prokaryotes. Yeah, I don't know whether you have heard about pangenomes. Basically, if you have 100 people in a room and you isolate E. coli from their guts, yeah, and you would sequence these E. coli's, only 5 to 10% of their genome would be identical. Not of the human, but of the E. coli, yeah. And the vast majority of the prokaryotic genome is actually encoded in pangenome. So that is where a prokaryotic bubble, if you want, can exchange DNA as much as they like. So bottom line is that no genome that you sequence today from a free-living cyanobacterium will ever match the plastid genome that was captured 1.8 billion years ago and fixed as a plastid. Yeah, you just have this gap in time, and you will never be able to really get there. Yeah, you can only try to get close, but you will never match it 100%. If you do an overall approach of complete protein homology, yeah, so then you end up here in section 5, cyanobacteria. What does this mean? Is just look at the outer bubbles. Yeah, you have this color gradient going from low to high genome identity. What was done here is, at the end of the day, you take the entire Arabidopsis coding capacity, yeah? So all of the proteins that are there in Arabidopsis, and you treat them as if they were one protein, yeah? One sequence. And then you align all of the cyanobacteria genomes to this one sequence as if it was one protein, and then just plot the overall identity, yeah? So you see that here in the section 1 cyanobacteria, which are the more simple cyanobacteria, so to speak, the homology is rather low. And once we move into these, yeah, that's also why it says here plastid ancestor, you just get the highest overlap in total sequence. Yeah, that is the picture that this paper plotted. And this also means that the plastid ancestor was not only able to fix CO2 through photosynthesis, but anyone that knows these cyanobacteria will also know that these cyanobacteria have the ability to also fix nitrogen. Yeah, so they are disotrophic cyanobacteria. Another paper, and actually more recent, a more classic approach, but probably done phylogenomics, they find that here the archaeplastida, so again red algae, green algae, glaucophytes, that their closest sister lineage in the cyanobacteria is here Gliomargarita. At that time when this paper was done that I just showed to you, or this work was carried out, Gliomargarita wasn't actually even known. Yeah? So this was part of the paper. They isolated this, it's a freshwater cyanobacterium, and came out to be the closest. So you can see that you've got these two models that pretty much contradict each other. On the one hand, here on the right hand side, cyanobacteria that can fix nitrogen as the closest ancestor when you plot overall homology. And if you do a more classic but up-to-date phylogenetic approach, then you end up in the section 1 cyanobacteria. So it's one or the other. So we actually, at this very moment, just do not know. Keeps to be excited. Plastid genomes. Basically all plastids, or the vast majority of plastids, they still have their own genome, yeah, for reasons that we'll get into later. Roughly 1,500 plastid genomes have been sequenced. Oh, I can actually put this in here. Live update. 1,500 plastid genomes have been sequenced. I checked that last week. It's really a rapidly growing number. And they vary in size dramatically. Yeah? So the smallest ones are only about 35 kb, so 35,000 base pairs. The biggest one that is fully sequenced, so where we have a full circle being sequenced, is 1.13 mega base pairs, so much bigger. But some plastid genomes, such as that of Acetabularia, which is actually a very big single-celled green algae. They are estimated to be about 2 megabase pairs, yeah, so quite big. All plastic genomes are characterized by an AT richness. Yeah, so you see that here at the bottom right in the sequence, this is what it means. Yeah, you all know that we have 4 nucleotides, and you either have AT or GC binding. And organelles, both plastids and mitochondria, they get enriched in AT coding, which is due to Muller's ratchet. Yeah, so up here in the right-hand corner, I just pictured a Muller's ratchet for you. So what this means is it's a one-way ticket in terms of mutations. Yeah, it's just easier to go from a GC to an AT mutation, and it's a one-way ratchet. Yeah, every time that moves that way, it can't really go back. It's been tested in the lab, and the chances of codon reversal, so going back from an AT to a GC, is actually 1 in a billion. Yeah, so it's very, 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 very rare, which is why they usually enrich in AT content, because they cannot recombine like their free living relatives anymore, because they are enslaved as an endosymbiont in a cell. We have a set of conserved genes present in all of the plastomes that have been sequenced, which is quite interesting, yeah, because you can probably imagine that they all went through different bottleneck events and different evolutionary fates, but somehow there's a core set of genes that gets constantly fixed, and we'll talk about that later, why that could be the case. If you look at plastic genome architecture, here on the right-hand side, this is a rough overview of what the vast majority of land-plant plastic genomes would look like. Yeah, you have a small and a large single copy gene region. This would be the single copy, and this would be the small, and this would be the large single copy gene region. And then you have these so-called inverted repeats, yeah, that you see here on the left and the right. They encode to a degree the exact same genes, and it's just the genetic arrangement that these plastic genomes have been fixed up on, and it's quite conserved across the land plants. But if you go into algae plastic genomes, you get, as you see on the right, quite a bit of variety in terms of both sizes and arrangements. But here, Cyanophora in the middle, and I told you earlier on that this is, if you want, the dinosaur among these lineages. You also get that rough structure that you see in land plants. So we do know that this general arrangement of plastic genomes is ancient, but there are some which are really wild here, Heterocapsa, and these are the dinoflagellates, and we'll get back to those because these are organisms actually going back to secondary endosymbiosis. One thing that if you study textbooks, yeah, and then you learn and learn and learn and learn, and you listen to talks such as mine, you know, that is the knowledge that finds its way into the textbooks. And but you need to understand that most research happens on very few model species, yeah. And here at the very bottom right, you have Arabidopsis thaliana. You all know Arabidopsis thaliana. It's what most of us in the plant world work with because it's so nicely accessible and so many tools are there. But you sometimes have to be careful if then knowledge from Arabidopsis is translated onto other species. Yeah, and that's what I want to communicate with this slide, because not all plastic genomes are the same. Yeah. So here, bottom left, you have the glaucophytes again, your dinosaurs, red algae, green algae, and then up here on the right, you have your embryophytes, so the land plants. And these little dots here on the radiation, what they communicate is just a few genes. We don't need, for today, it's not interesting what these genes are, but you can see that, you know, with ongoing evolution and the host taking over more and more control over the endosymbiont, yeah, so over the plastid, more and more genes are actually lost, yeah, either lost entirely in evolution, or they get transferred to the host nucleus for control purposes. So just always keep in mind that whatever lineage you sample, don't translate it to 100% to all of your other species. Yeah, and this was actually important for us, and this was actually part of a paper on photosynthetic slugs, but that would be a different talk, where we worked on the plastid biology in these animals, and it really was important because some of these slugs feed on red algae and some feed on green algae, and that makes a big difference in terms of plastid survival in animals. So I just mentioned earlier on that basically you always find a core set of genes encoded, by the plastid genomes, and the same is true for mitochondria. If you look at all our mitochondria, they encode, I think it's 13 to 16 proteins or so, and that is it's rather conserved. And the question, of course, is why don't all genes get transferred? I mean, we have 1,000, 1,500 proteins easily encoded in our nucleus. They get targeted to the mitochondrion. Plants, same thing. You have 1,000 to 2,000 proteins in the nucleus encoded, which get targeted to the chloroplast. So of cyanobacterial ancestry, where you have gene transfer from the chloroplast to the nucleus, and then the protein getting re-imported. Why not all of them? You'll find quite a few different ideas popping up in the literature. One of them is hydrophobicity, so that some proteins are just too hydrophobic. So they're too big and cannot be crossed across the membranes because they're too hydrophobic. Some say, well, the product is actually toxic in the cytosol. So imagine gene is encoded in the nucleus, gets translated in the cytosol, and needs to be targeted. This intermediate step in the cytosol is toxic, and that is why it's genetically selected against. Genetic background compatibility, so that there is the genome background of the chloroplast just doesn't match the nucleus for these genes for some reason. Limited transfer window hypothesis, so that's, that evolution only has a certain available time to pass the genes, and then at one point the door just closes, and that's the end of the story. Or complex assembly of subunits is another point, so that you would only be able to transfer all the genes all at once because they all need to be assembled at the very same time, and that would just complicate the matter. We now know that a few of them are very, very unlikely. Hydrophobicity is one of those that still is a strong competitor for the reasons why not all genes have been transferred. One that I really like, because it nicely explains it for both organelles at the same time, is the so-called co-hypothesis. So this is the co-localization of redox regulation. We don't need to understand the biochemistry in detail for today. It's a model by John Allen. But the idea is that the biochemistry that's happening at these membranes, so whether it is in the mitochondria for ATP synthesis or in the chloroplast and thylakoids, the photosynthesis, that you need a direct regulation of gene expression. So at the end of the day, if we go back here, something happens in the chloroplast or mitochondria, it needs a new protein, and the communication pathway to the nucleus and sending back the protein would, at the end of the day, take too long. So if you co-regulate redox regulation with transcription and translation of the proteins, it would explain why you have this feedback loop, so the genome being here directly where the proteins are needed. So it's an elegant model, and if you want to, you can read up on it. But it would explain why mitochondria and plastids, so all of these which still photosynthesize or respire, still have genes encoded. Another interesting thing in terms of the endosymbiotic phase that plastids have taken is that not all plastids, share the same biochemistry, and not all lineages took the same phase in terms of how they decided what to do with all the biochemistry that came in. So here, just to show you examples: heme biosynthesis, starch biosynthesis, aromatic amino acids, and some lipid precursor biosynthesis. So if you imagine the endosymbiotic event in green, you would always have your cyanobacterium. Early on, we know that the host as well as the cyanobacterium were able to synthesize heme. And then all of the lineages picked this here, so it must be a very ancient solution. That the chloroplast now synthesizes heme and sends it to the mitochondria. This is the case for all phototrophic eukaryotes. Starch biochemistry, two different pathways. So we had an UDP and an ADP dependent pathway, the ADP being the cyanobacterial version, UDP to glycogen being the host version. And if we now look at the red lineage, they actually decided to stay for some reason with the original host cell version, and they make fluoridine starch in the cytosol, not in the plastid, whereas the green lineage, the chlorophytes, like the land plants and all the crops that we eat, at the end of the day, they have picked the cyanobacterial pathway and deposit starch in the plastid. Yeah, here aromatic amino acids or lipid precursor biosynthesis. Yeah, just two other examples. Again, here you will throw out an entire pathway from the host and just keep the cyanobacterial pathway. But this is also not always the case. As you can see here, you can also sometimes just see that both are still present in the host version and the cyanobacterial version. But very often, and this is true for a dozen or so other pathways, the cyanobacterial version appeared to be the superior version and has replaced the ancient eukaryotic host cell version. One aspect of plastid origin is the so-called monoplastidic bottleneck, and this is something that we are currently also still working on, and which I find both genetically as well as cell biologically quite attractive. So what does this mean? Just all follow me through this picture here. We start at the bottom left. So that would be the origin and the very first steps of integration. Yeah, so you have some cyanobacteria and a phagocytosing host cell. They could have been epibionts, and that is how the symbiosis began. Maybe it was just really cyanobacterial prey to begin with, but there were multiple. This is the point. Yeah, and at this point, you had a free-living cyanobacterium that was dividing by itself and had its own biology, and you had a host that was dividing by itself. Yeah, so it was a pre-synchronized phase. They were both dividing independently. Once you get to the endosymbiont stage, yeah, you now have cyanobacteria that are dividing in the host cells. And what EGT means is endosymbiotic gene transfer. So this is the point where genes are being transferred from lysing cells, lysing cyanobacteria, to the nucleus. And we know also from empirical studies in the lab, actually, that the more endosymbionts you have, the easier endosymbiotic gene transfer is. Yeah? And this is also easy to understand, because if you only have one plastid and it breaks, you're dead. Yeah? If you have multiple plastids and you can remove damaged plastids by an autophagosome, but the DNA can then freely float over into the nucleus, but float over into the nucleus, be integrated, it's understandable why multiple endosymbionts just, make it easier for the endosymbiont to be established. And you get different genes transferred. This, for instance, FTSZ, division gene, and so on and so on. But what we then see is that, and this is true for all of the algae lineages, and Jan at that time, he looked at 180 or so different ones where we knew the morphology of the species as well as the plastid genomes, is that the basal branching ones, so yeah, both in the red, the glaucophytes, and the green lineages, always the bottom ones, they basically now have one plastid that is dividing per nucleus. Yeah, so here you still have a non-synchronized division between endosymbiont and host, where now you go through this monoplastidic bottleneck, and which we believe was or is associated with synchronizing the division events. Yeah, so it's just easier if now the plastid can send one signal to the nucleus, and this has also been molecularly analyzed. We do know that there is a signal that goes from the plastid endosymbiont to into the cell cycle of the nucleus, and that says, look, I'm ready for division, you can go. Yeah, and that's why both in the red and the green lineage, we see monoplastidity being actually the main thing that you observe. So if you look at algae in the oceans, for instance, yeah, most of them will have one plastid per nucleus. But at one point, it got desynchronized again. Yeah, so both in the green and the red, both in the red and the green lineage, we know that through gene losses or replacement events of, in particular, genes associated with plastid division, which makes perfect sense, we now get back to multiple plastids per cell. Yeah, so now plastid division is again decoupled from nuclear division, the same way as it was very early on during evolution. And this is, it's interesting that evolution had to go this long path, but it's also interesting to know how important this was, because we do know that both in the red and the green lineage, this was actually associated with macroscopic body plants. Yeah, so if you're thinking, if you go out to the oceans, also in Brazil, and you go diving, you know, all of the big red kelp that you see, they also all went through this monoplastidic bottleneck, and they all have multiple plastids per cell. And the same is, of course, true for land plants. And we do know that this is associated with the transition from water to land during land plant evolution, because some of the basal lineages that we're working with, or the algal sister lineages, I should say, to land plants, they have 1 or 2 plastids per cell, and it's also tightly synchronized. And we, however, have some mosses, such as Marchantia. Some of you have probably heard about Marchantia, and it's a really nice model system in the lab to do evo-devo with. In Marchantia's life cycle, they actually have in the spores 1 plastid per spore and per nucleus, but then the moment that they start to germinate, they leave this bottleneck again, and they become polyplastidic, to grow into their macroscopic phenotype. And when they make new spores, they again cease plastid division and pick 1 plastid per nucleus for the spores. So they go through this cycle as part of their life cycle. It's quite amazing to watch. Another thing why multiplastidity is important is it allows to diversify plastid types. Yeah, if you only have 1 plastid per nucleus and 1 plastid per cell and 1 plastid type, there's not much you can do with it. Yeah, because it's always the one that has to do photosynthesis. If you're thinking about a land plant, yeah, and if you're thinking about the many crops that we eat, yeah, think about a carrot. Yeah, most of the plastids in carrots, they do not photosynthesize anymore, really, or that's definitely not their main job. But they're there to store starch, yeah, in terms of aminoplasts. You have ethioplasts, you have chromoplasts, yeah. If you're thinking about all the beautiful flowers, they basically just enrich some pigments to become more attractive to pollinators and so on and so on. So all of these different plastid types, they are also unique to land plants, yeah? And you can only evolve this if you have escaped the monoplastidic bottleneck, which I think is quite fascinating. If you think about the origin, it was multiple cyanobacteria, and then you get a million years of evolution. Most algae still only have 1 plastid per nucleus, and then it goes escapes this bottleneck, conquers land, and evolves all of this beautiful flowering complexity that we see today. And that takes me to secondary endosymbiosis. So the almost last part of the talk today. Secondary endosymbiosis is a whole different story and was way more complex than primary endosymbiosis, and it also happened multiple times, as you will see. If you're looking at the tree of life again, yeah. So this here is a tree of eukaryotic life, so to say. These are all the modern eukaryotic groups that we know today. We do see here highlighted with these stars that phototrophic eukaryotes are paraphyletic. Yeah, so we find them among Rhizaria, Straminopiles, alveolates, haptophyte algae, cryptophyte algae, and of course the Archaeplastida that I talked so much when we talked about primary endosymbiosis. So all of these eukaryotes, they're not monophyletic per se, of course, here at the root at some point, but they are paraphyletic in terms of their ability to photosynthesize, whereas the plastid that allows them to photosynthesize is monophyletic. Yeah, so this emerged only once in the Archaeplastidal lineage. And secondary endosymbiosis is, at the end of the day, what explains this discrepancy, if you want. Yeah, so on the one hand, this doesn't make much sense if you know that the plastid is monophyletic, but the moment that secondary endosymbiosis comes into play, it all falls into place. And this is only 30 years ago or so that became clear why that is the case. So what is secondary endosymbiosis? The one take-home message, for today, maybe, if you want to, have a clear cut between the two, is primary plastid endosymbiosis. Primary endosymbiosis is a prokaryote and a eukaryote. Yeah, so you have a eukaryotic heterotrophic host that engulfs a cyanobacterium and evolves it into a plastid. Secondary plastids and secondary endosymbiosis is a eukaryote and a eukaryote. Yeah, so both prey and host are eukaryotes. Yeah, so again, a heterotrophic eukaryote, but in this case not engulfing a cyanobacterium, but another algae that already went through endosymbiosis. And this happens simply because protists feed on protists. Yeah, these are beautiful pictures from, I think it's the Nikon competition that gets called out every day. So here, bottom left, you would have something like tetrahymena, yeah, but definitely ciliates. And you can see they are big, beautiful critters, and you can see that their cytosol is basically full of both prokaryotes and eukaryotes that can photosynthesize. Yeah, so it's just feeding on all of this, and it's then well, not easy to imagine, but definitely one can imagine that at one point one of them gets stabilized and evolves then into a phototrophic organelle. And here you would have an amoeba. This is, I think, actually, this is chaos, I believe. If you ever get to see them under the microscope, it's fantastic to watch. And this here is, as you can probably see, already a big eukaryotic algae, and you can see how big chaos is. Yeah, and they just really engulf all of its prey. And then again, they end up in the cytosol, and that is how you get a eukaryote established in a eukaryote. And this is easier for several reasons. When I talked about the monoplastidic bottleneck, when you think about it, when you establish a prokaryote and a eukaryote, you also mix prokaryotic biology with eukaryotic biology. You mix a prokaryotic, not really existing cell cycle with a eukaryotic cell cycle. Yeah, so homogenizing these two biologies is just way harder, most likely, than merging two eukaryotic biologies, yeah, that are just more familiar with each other. And that is probably why secondary endosymbiosis has occurred so many times more frequently than primary endosymbiosis. Secondary endosymbiosis is interesting because it really provided us with a massive amount of very, very interesting supergroups, yeah, and quite a long list of them. So here, this is just the things that you've already seen. Again, at the top left, you have primary endosymbiosis, and to recapitulate the three lineages, glaucophytes, rhodophytes, and the chloroplastida. And then here on the right-hand side, secondary endosymbiosis, where independently red algae was engulfed by a host, red lineage, secondary red lineage. And then here independently a green algae was engulfed, and you get secondary green endosymbionts. Yeah? So we do know that this happened multiple times independently. And here at the bottom, this is what I mean by the intriguing supergroups that have evolved. Yeah. So these four here, they are all part of the red lineage, and that's what I meant earlier when I said the red lineage gets interesting when we delve into secondary endosymbiosis, because what you have here, for instance, are the haptophytes. So anyone who's ever taken a ferry from, and I don't know how many from you in Brazil have taken the ferry from France to England, yeah, but if you then see these big white cliffs of Dover, you can also look them up online, yeah, so it's these 5 to 7 meter high cliffs which are just entirely white chalk. All of this chalk is basically a deposit of haptophyte shells, yeah. So they are very important primary producers, and were so, especially in the ancient times in the oceans, yeah, providing tons of food, and were really a very successful lineage. Heterocontrophiles, so straminophiles, diatom algae, yeah, they also belong to this group. Also, again, very important because they're substantial marine primary producers. So they stand at the very first parts of the food chain, yeah, and they are also the ones that are really struggling with global warming and an acidification of the oceans. So that's damaging them, and it's actually quite important to monitor them. And from what we can tell, unfortunately, a lot of lineages have already been lost, and they're really truly suffering. But heterocontrophites, important ecologically as primary producers. Apicomplexa, a very interesting group that I'll get to at the very end to wrap it all up. Because this group is entirely composed of parasites, yeah. The most famous one being the malaria agent, yeah, so Plasmodium, but also Toxoplasma gondii, for instance, yeah. They all belong to this group of Apicomplexa. So what some of you might not have known, or most of you might not have known, is that the global killer #1 still of humans, next to humans themselves, so Plasmodium, which causes malaria, was actually once a free-living red algae. Yeah, but this entire group also of red secondary endosymbiotic origin. And then dinoflagellates. The next few slides will be on dinoflagellates because they are just amazing in terms of their biology, and they are infamous for causing red tides, which are also increasing globally because of global warming, because they just love it. And that's when these algal blooms occur and you see these red tides, very often caused by dinoflagellates, some of which are actually toxic. And then here from the green lineage, the most famous ones would probably be the eugenophytes for two reasons. One is that they're interesting in terms of biotech, because they make very interesting wax esters that you can use downstream. So that is where they are being analyzed for. But they were also the ones that were under the microscope when Sarah Gibbs in the 1980s, I think it was, when she saw these cells and saw the plastids, so the endosymbionts, and she noticed that they have multiple membranes, so more membranes than the plastids from plant cells. And she then basically formulated the idea that maybe what we see here is an algae inside a eukaryote. So she was the mother of the secondary endosymbiotic world, if we want. So that was based on observing euglanophytes under the microscope. It's also interesting for anyone that loves genetics or cell biology, for the simple reason that secondary endosymbiosis has generated us, or has provided us with the most complex eukaryotic cells that we know. Two main reasons being that, first of all, we now have cells that contain 4 different and independent genomes, in some cases. So in Gouldardia, red lineage, Bigavella, green lineage, what you have is, of course, because it's a eukaryote, you have the nucleus, same way that we have it, with several 10,000s of genes. Then you have the mitochondrial genome, also the way that we have it, of course, as a eukaryotic cell, with a few dozen genes. And then you have here the endosymbiont, and in these cases, the former nucleus of the endosymbiont was actually also retained and reduced. It's not fully vanished. It's called the nucleomorph, and they encode a few hundred genes and different sets in both the red and green lineage, but it's still conserved. And this was also, when these nucleomorphs were identified and sequenced, this was the last remaining evidence that was needed to prove that these endosymbionts are of eukaryotic origin and not cyanobacterial origin. And then, of course, at the end of the day, you have the plastid genomes in both of them, so 4 genomes. The other reasons for complexity is the multiple membranes that surrounds these organelles, So the plastids of these organisms are always surrounded by a minimum of 3 membranes. The majority have actually 4 membranes, such as these two. Both Bigavella and Gouldardia would have 4 membranes. And that makes targeting of proteins, which are nuclear encoded, really problematic. And that was, as was mentioned earlier in the kind introduction, also part of my PhD studies, actually, to investigate how nuclear encoded proteins find their way across the membranes into the plastids. So really intriguing in terms of the cell biologic nature of these critters. So fascinating to study, really. And nature didn't stop at secondary endosymbiosis. The title of the talk stops at primary and secondary endosymbiosis, but nature didn't. And it's actually the dinoflagellates that I mentioned earlier, which are the true champions of doing so for some reason. It's not exactly understood why, but I'll show you some, just in 2 slides, why that could be the case. But it's only an idea. We don't know why it is. Dinoflagellates are beautiful. They are also the ones that I mentioned earlier, Ernst Haeckel and his office that you can still visit in Jena. And dinoflagellates is what he really loved to paint. This is, of course, not a Haeckel painting. I should have put one in. These are just microscopic images, but they make these beautiful, intricate shells for many, many different reasons. And there are hundreds and hundreds of different versions. But what dinoflagellates also do is they actually incorporate algae which go back to secondary endosymbiosis. So here, if you want, this would be primary endosymbiotic level, and then the next level is secondary endosymbiosis, which gave us the organisms that we already talked about, The dinoflagellates here, Dinophyta, they then engulfed, for instance, a heteroconophyte or a haptophyte or a cryptophyte. And from that, we get different new eukaryotic lineages, which are now tertiary endosymbionts. Yeah, so it's a bit like the Russian Matoshka doll, where you have one doll and the next and the next and the next. And this actually goes up. I think currently microscopic evidence exists for the 5th level. Yeah, so quite amazing, if you want. And dinoflagellates just mastered this. I already mentioned red tides, for which dinoflagellates are sometimes known. I actually should have looked it up. I don't know about the situation around the Brazilian coast, but I'm pretty sure you also have red tides. What you see here, maybe you can see this usually with the audience. I would ask whether they know where this is on the globe, but I can't do it today, so I'll just tell you. This here is the UK, so this here would actually be the channel. So you would have Europe sitting here at the bottom right, and then here this is England, the channel, and this light blue coloring that you see, this is algal bloom. So these are red tides. And I only put up this picture so that people get an idea of how big these blooms actually are. Because if you're standing at the beach and you see these waves flushing in, very often, unfortunately, with tons of dead fish because of the toxins that these dinoflagellates secrete, you don't really get an idea of the sheer biomass that generates with these algal blooms. Here at the bottom right, this would actually be Germany down here. This here is Norway, covered in snow, and then Sweden on the right. So again, in the bottom left part of the picture, and I think you can see my mouse cursor, that is, again, we would have an algal bloom basically covering the entire coast of the Netherlands. Here you would have all the little islands that I sometimes visit for holiday reasons. So this is just a massive amount of algal bloom. And in some countries, when you go to the beach, you will actually get signs such as this, raising algae awareness. And then they have like a flag system, and at a certain level, you shouldn't enter anymore. And most of the times, it's actually dinoflagellates that cause this. A more prettier sight, and I hope some of you had a chance to experience this live in nature, because it's amazing to watch, is bioluminescent. So these pictures are not manipulated, that they are, from what I have been told, that's exactly the brightness that you then actually see. I've had it myself, walking along the beach, and it really is quite bright. But that's also what dinoflagellates do, and it's basically a signaling mechanisms among them. So they have this bioluminescent feature, and that often are also dinoflagellates that can do this. So why are dinoflagellates so successful in incorporating other protists? Well, going back to this picture, I showed you that many of them have these intricate shells. And of course that confines the size that they can phagocytose. Because they have a rough and stiff shell. So and some only have these small tunnels. But for that reason, they have actually evolved extreme feeding modes. The so-called paleo, which is at the end of the day just an extension of their entire cytosol, which they can secrete around other algae or prokaryotes. And for them, it's a way to get around their restricted cell size. And it's quite amazing. This here is a picture from Jacobson and Anderson from the 80s on Protoperidinium. It was just a time where most of the great microscopy was done on these critters. And they observed that basically at the end of the day, this entire prey diatom was engulfed by the cytoplasm of dinoflagellate. And here you can see it in a live shot. And you see maybe here this pallium really wrapping around. In this case, I think it's a cyanobacterium. And it's just the way how they get around the restriction of their cell size by just secreting the entire cytosol and a membrane around their prey. And then they slowly digest it and can take up entire chunks. So quite amazing to watch. Genetically, they're also interesting because, and I hope for some of you this is new knowledge, because that's the main thing of today, of course, is that they have some of the biggest genomes. Here on the right, what you see, this is a scale of genome sizes. So up here you would have prokaryotes, gram-positive bacteria, gram-negative bacteria, fungi, algae, worms, insects. Where are we here? Mammals, yeah. Big genomes, but not very diverse, so quite restricted in genome complexity sizes, but big. Amphibians, and then flowering plants with their massive genomes, because what they have is lots of whole genome duplications. That is one of the main reasons why they have such huge genomes. But down here, it wasn't part of the scale. I added dinoflagellates, and the genomes that, from what I know, have been mapped are 1.5 to 185 gigabase pairs. So they are massive, yeah. And not only do they have the biggest genomes, they also have very specialized chromosomes, because they're permanently condensed. So if you maybe remember from biology classes, if you think about mitosis, meiosis, our DNA is more or less fluffy, floating around, and only for mitosis and meiosis do the chromosomes condense for the division process. But in dinoflagellates, for some reason, that is the permanent state. Yeah? So they are always semi-crystalline. It's amazing to watch under the microscope. And it's just probably the way how they arrange these massive genome sizes. And this is also always a nice reminder at the end of the day that genome size just has nothing to do with with organism complexity. It might be a simple message, but it just every now and then pops up, especially when you look at some work on the human genome where every nucleotide is interpreted. Genome size is not coupled to organism complexity, yeah? And you can probably not see this properly on your screens, but maybe downstream in the video. But you also don't have to read the species name for today. But each bubble is plotted correctly in terms of genome size. Yeah, so the bigger the bubble, the bigger the genome. Up here, this green bubble, this would be Zea, mice. Yeah, here you would have Zygnema, green algae. The little bubble here, so top middle, this is Arabidopsis, what we already talked about. And then the bigger blue bubble, bottom right, yeah, but where you see the full circle, this is the human genome, yeah, and then at the bottom, that would be an amoeboid genome, yeah. So this is a single cell, like the cow's that we just saw, so multiple times bigger than the human genome. And then here in red, this would be, dinoflagellate genomes. Again, way bigger than the human genome. So the biggest genomes, but not coupled to organism complexity. Another peculiar thing which is associated with their condensed chromosomes and their big genomes is that among eukaryotes they are unique in terms of their histones. So most of them encode no or very weird histones. Yeah, so here it will always be a dinoflagellate histone mapped to the human version at the top, H2A, H2B, and so on and so on. But the take-home message is that these don't really look functional, and we don't exactly know what the histones in dinoflagellates are doing. But it's a unique feature because it usually is the one thing that all of the eukaryotes also have conserved. But you can always, if there's in any test a question of, what eukaryote is different in biology than any other eukaryote, dinoflagellate is most of the times the correct answer. But what happened, if they don't have histones anymore and use that for DNA organization, well, what do they do? Well, that's, again, an interesting evolutionary thing that occurred, and that is they probably replaced it with a viral protein. And this was a great work by Sebastian Gornick some 10 years ago. So they encode no histones, but they have this weird nucleoprotein which you otherwise only find in viruses. Yeah. And so here this would be a sequence alignment. So if you go to any sequence database and you search, you only find these sequences in dinoflagellates, in all of the dinoflagellates, and in a group of viruses that is also known to infect dinoflagellates. Yeah. So it's this DVPN proteins, dinoflagellate viral nucleoproteins. And they've also localized it, and they can see that this protein co-localizes with the condensed chromosomes, and that it is probably involved in chromosome condensation. So somehow this viral protein has taken over that function and provided dinoflagellates with this unique feature. So what this means evolutionary is, so this would be a very simple cladogram, ciliates, apicomplexa, Purkinsus, and the dinoflagellates up here. So these are all alveolate organisms. What happened is a viral infection at the base of the dinoflagellates, and that actually then by a virus, and that gave rise to the dinoflagellate group. Yeah, so quite amazing. One last thing where dinoflagellates are unique is also the way how they encode their plastic genomes. So this picture here on the left is what we saw earlier. And we are already mentioned down here, these little circles, heterocapsa, that dinoflagellates do differently. And that's what you see here on the right. They actually encode their plastic genomes on so-called mini-circles. What that means is each gene, roughly, is encoded on one single small mini-circle. Yeah, so they don't have one big circle with all genes, one circle, one gene. Yeah, that's, again, something where dinoflagellates are just unique. And that takes me to the add-ons. We're now an hour in. The question is, do you guys want a break? Type into the chat.
Alguém acha que seria legal intervalo aí?
It's 20 more slides or so. I don't care. It's up to you.
Maybe 10 minutes.
Okay.
Okay. Trend health here.
Yeah.
Okay. We come back. Welcome again, people. Okay,
Hello.
we have the words.
I have the words. Excellent. Okay. So everyone back. Thanks for your patience so far. I hope you enjoyed it. I'll basically wrap up with one story that we're working on now. It takes us back all to the origin of life, and then at the very end we'll just look at some interesting biology among parasites, which were once free-living algae of secondary endosymbiotic origin. So the slide that you see here, something I published a few years ago, where I tried to get in all of the evolution of the main import or protein translocation machineries that most of you, or some of you, will be familiar with into one slide. Because one thing that is evident is that endosymbiosis precipitates targeting evolution, yeah, because any endosymbiont, we talked about it early on in terms of the origin of eukaryotes, so the mitochondria, any endosymbiont that comes in requires new targeting pathways. So to go through this picture here, at the bottom you would have the hydrothermal vents that I mentioned earlier on in the origin of life, yeah, and there are a few components only which are conserved. And this dotted line basically pictures the plasma membrane, where we don't know what was there at the beginning, yeah, but we had 70S ribosomes and already a gradient at some kind of intersurface for ATP synthesis, yeah, so the very core of life, if you want, and that is ATP synthesis and an energy household. And then when evolution commenced on to the next level, and that is, provide us with the first free living cells, in blue the bacteria and in red the archaean. This is already where we had many membrane sitting translocation machineries, the SEC pathway, which is universal, the TAP pathway, ATP synthase, and so on and so on. And here the type 1 to 4 secretion system in the gram-negative bacteria. Yeah, but membranes need translocation machineries. Then in eukaryogenesis, when these two fused, so this is the endosymbiotic event that gave us the eukaryotic cell, we get different fates of the different translocation machineries. Yeah, so some have actually persisted in the mitochondrion, both in the outer and the inner membrane. They were remodeled and they were complemented with many eukaryotic specific components in the TON and TIM machinery. But quite a lot have maintained themselves in the mitochondria. Some were recolocalized and reused in the host cell, such as the here in red, the SEC-YEC machinery of archaeal origin, which now in us eukaryotes is known as the SEC61 machinery in the endoplasmatic reticulum for protein import into the ER. Yeah, so we have co-translational import. But endosymbiosis means the evolution of translocation machineries, because what endosymbiosis does is it provides you with new membrane compartments inside a cell. And it all gets a bit more complex if we then move on to secondary and primary plastid endosymbiosis and secondary plastid endosymbiosis. Sorry. So here at the bottom, yeah, this is now the plastid came in, and you have a whole new set of import machineries. And surprisingly, many of those actually look very much like what you see in the mitochondria, probably because both of them were gram-negative or belong to the gram-negative bacterial group. Yeah, here alpha protobacteria origin, here cyanobacteria origin. But at the end of the day, very much alike. Secondary endosymbiosis, so here now where you have the eukaryote sitting inside a eukaryote, in some cases, and that's why it looks so strange, these algae cells actually sit within the endoplasmatic reticulum of the host cell. So a very strange arrangement. But this is the case without a doubt. And that's what I meant a few slides earlier, that they generate the most complex biological cells, because you have all these multiple membranes now separating the cytosol from the plastid stroma. So all the membranes need to be crossed by proteins. Yeah. So if you have a thylakoid protein, for instance, it's 5 biological membranes that need to be crossed by a protein. Yeah. So always keep in mind, endosymbiosis, it adds new layers of membranes, and each new layer of membrane means a new way of how proteins and any other substrate needs to be translocated. And it's, for an evolutionary biologist, quite peculiar that both the mitochondrion and the plastid have chosen such identical pathways for import. Yeah, a lot suggests that the machineries you see in the mitochondrion on the left, and please don't try to get into each and every protein today, that's not the point of this slide, it is just the way that it is, and they are complex machineries. The point is to see how similar they are. Yeah. So here in this yellow-orange tint, you have the mitochondrial input machinery, and on the right in green, you have the plastid chloroplast machinery. But both probably originated to a degree from protein secretion machineries. So when they were free-living bacteria, they were secreting proteins through these channels, and then the genes migrated from the organelle to the nucleus, and then they were re-imported, and that also changed their orientation so that they were now integrated from the cytosolic side and were serving protein import. But both look very similar, both use receptor platforms, both use N-terminal targeting peptides that you might have heard about to be recognized in the cytosol and then imported into mitochondrial matrix and plastid stroma. So how is mistargeting avoided? How does the cell know that a chloroplast protein is not a mitochondrial protein and vice versa? Yeah, because we know that 1,000 proteins or more are very specifically imported. And we also know that the targeting information is quite similar, but what it looks like is that one of the main denominators for the right localization appears to be the charge in the targeting peptides. So imagine a targeting peptide being like a postal address. Yeah, so it's a postcode on the proteins so that the organelles recognize. Yeah, so it says, oh, mitochondrial protein, oh, plastid protein. And what you see here in the middle, this is just the first 20 amino acids of an Arabidopsis thaliana protein. Yeah. And this protein, I believe it's a phosphatase, but that's not so important for today, is dual targeted. Yeah, so the genome encodes one protein. Yeah, there's one gene, one protein gets translated in the cytosol. It has an average charge. So if you take all these 20 amino acids and you do, positive charges, negative charges, and you do an evaluation of 2, yeah, plus 2. But it's dual targeted. So 50% of the protein goes to the chloroplast, 50% goes to the mitochondria. Evolution then picked a different path, and I gotta go into full screen mode now. For a rice protein, yeah, in rice we actually get 4 copies in the genome, and 2 of which get targeted to the mitochondria and 2 get targeted to the plastid. Yeah, and you can see on the right hand side that if you look at the overall charge for those proteins that get targeted to the mitochondria, it's plus 3.5, and those that go to the plastid, it's plus 1.5. So evolutionary, a completely different way how to solve the problem. Yeah, the Arabidopsis protein has an intermediate charge, goes to both organelles, and rice, Ovoza, just encodes multiple copies with different charges, and half go to the mitochondria and half go to the plastid. And a group in Australia, they went a step further, and this is now for Plasmodium falciparum, the malaria agent that I'll be talking about in a few slides. They made synthetic peptides, yeah, and one was called Skinny Slinky Kinky Thing, yeah, and this had a charge of plus 4.5 and went to the plastid. And then they made one which had a negative charge of minus 2, and it also read, it will never target plastid, and it didn't. It got stuck in the ER and never found its way to the plastid. So what this demonstrates is that the charge in the very N-terminal sequences plays a very, very important role in the localization of these proteins. Yeah? And when you take all of the nuclear-encoded proteins in Arabidopsis that are targeted to the plastid, yeah, and you squeeze all of the sequences for, in this case, plastid proteins, in the middle dual targeted proteins, and in the right-hand side mitochondrial proteins, and you do these so-called sequence logos, what you get is that the plastid proteins in Arabidopsis, they have an overall charge of plus 2, Yeah, and the mitochondrial proteins, they have, like our proteins, yeah, so our nuclear-encoded mitochondrial proteins, they all have very highly positive charged N-termini to be targeted into the mitochondria, which has to do with the inner membrane and delta psi. But point being, the ones that are dually targeted in Arabidopsis, so here the middle plot, yeah, in orange, they have an intermediate charge that sits right in the middle between these two. So it's an evolutionary compromise, yeah, that if you want to have, or if you only have one gene, but you want to target the proteins to both organelles, you have to, at the one hand, make the mitochondrion happy. That needs a very positive charge for targeting recognition, but also not too much, because then it wouldn't be able to find its way into the plastid. And evolutionary, and now I'll go into full screen mode again because it's 2 slides. Evolutionary, this is a bit of a noisy slide. There's a lot to digest, but I'll guide you through it. What we see is that at the top right, actually, I'll do it like this so you have a mouse cursor. At the top right, this would be the origin of the eukaryotic cell. Yeah, so the mitochondrion comes in, you evolve your normal heterotrophic protist. And proteins that are then targeted from the nucleus, translated here, and into the endosymbiont, they need a very positive charge. Yeah, so just like what I showed you here for Arabidopsis, this big blue bar, yeah, high positive charge. So this is like the charger on your mobile phone indicator. Normal eukaryotic cell, think about any cell in your body, yeah, it would look like this. Nuclear encoded, translated in the cytosol and targeted to the mitochondria, very positive charge. Because you need to cross the inner mitochondrial membrane, which is highly positive charge because of ATP synthesis, as you probably know. Now, plastid acquisition, so primary endosymbiosis of the cyanobacterium. Now you have nuclear-encoded proteins that can be targeted to 2 organelles of endosymbiotic origin, either the plastid or the mitochondrion. If you want to go to the mitochondrion, that's what's shown here at the lower part, again, like you have in any heterotrophic protist or our cells, high positive charge. If you want to go to the plastid, low positive charge. Yeah? And those proteins that get dually targeted, so one gene translated and then off 50% to the plastid, 50% to the mitochondrion, you have an intermediate charge. And then on the right-hand slide, so forget what you see on the left and just look at the right-hand slide. That's basically what we get here. This is now secondary endosymbiosis. Yeah? So now you basically take this eukaryotic algae and you put it into another eukaryotic heterotroph. You have your normal canonical mitochondria, positive charge. Now you have nuclear-encoded proteins that are targeted to the plastid, intermediate charge, like you have it in your plant cell. But because you have this very special situation, and I showed you that earlier, and if it's okay for you, I can quickly go back to this slide. Yeah. So if you remember this one, where I introduced these the most complex cells that we know, I mentioned that there's a remaining nucleomorph. Yeah. So this is the former nucleus of the algae that is still present, and this also still encodes proteins that are targeted to the plastid. Now, what does this mean for the charge evolution? Yeah, if this nucleus is actually separated from the mitochondria. Yeah, so really think about this in room terms. Yeah, this nucleus here of the former algae sits in a different cytosol than this mitochondrion. Yeah, and what we actually see then in evolution is that now these former nuclear, so nucleomorph encoded plastid proteins, they can also have a positive charge, the same way that the mitochondrion actually has. Yeah, so what we would see here, but which is not allowed in plant cells. I know this is very complicated. Just look at the video again. It's difficult to communicate through this medium. In a room it is easier. But why is this the case? Well, because here the plastid endosymbiont, so this organelle, sits inside an independent cytosol surrounded by additional membranes, so there's no evolutionary selection pressure on the charge, because it cannot be confused with a mitochondrial protein. Yeah? There is no mitochondrion here in this cytosol, there's only one here in the cytosol of the host cell, not of the endosymbiont cell. So now evolution allows these sequences to also evolve positive charges because it doesn't interfere with the mitochondrial import. The way that we see it here in, for instance, an Arabidopsis cell where plastid endosymbiont and mitochondrial endosymbiont are on the same cytosol, yeah, where you have to, or where you have selection pressure on these charges. You do not see that anymore. And so this is a nice way how evolution, and again, we're looking at 2 billion years of evolution here, where you see how these 2 endosymbiotic events led to 2 different organelles using very similar import machineries, and then evolution coming up with different methods, and here mainly being the charge differences, to select between these 2 endosymbionts where the protein should be going. And once evolution goes a step further and separates or wraps another membrane pair around one of the organelles, selection pressure is released, and now it can look like the mitochondria. So it really it closes the circle in terms of an evolutionary endosymbiosis. And to wrap up the targeting part, what's really nice about the whole plastid endosymbiont evolution is that here the import is quite nicely studied. And what we see is, if you look at the left, so on the right-hand side, we know most of the individual components that now play a role. Actually, a few have been added since this review was published by Becker and Schleif. But here on the left, what you see is at the bottom line, your cyanobacterial situation. Yeah, so you have an OMP85 protein. This is quite broadly distributed among many prokaryotes. It's a protein secretion machinery, so it sits in the inner membrane of gram-negative bacteria, and it recognizes proteins to be secreted in cyanobacteria through a phenylalanine amino acid in the C-terminus. Yeah, so it's a single amino acid motif, gets recognized, and the protein gets secreted. Then, when the cyanobacterium became an endosymbiont, yeah, and a plastid, for instance, in glaucophytes, the whole machinery was turned around, yeah, because now the gene is no longer encoded in the endosymbiont genome, but in the nuclear genome. And the translocon basically gets integrated from the cytosol into the membrane, and now it's an importer. It's no longer a protein secretion machinery, it's protein import machinery. But it has retained also the targeting information. So we know from hundreds and hundreds of glaucophyte proteins that are nuclear encoded and plastid targeted that they have an N-terminal phenylalanine. Yeah? So same mechanism, but different orientation. This is also still conserved in the red lineage, so also rhodophytes, thousands of proteins actually across many species identified with this conserved residue for import. This changed then in the green lineage. So the main importer, which evolved from OMP85 to TOX75, is still there, but in the green lineage we get a duplication. Yeah, so here we have only one pathway, so it's like, imagine any road that you can think of. Yeah, it's a one-way road. Yeah, proteins can only be imported, and all cars and all trucks and all trams, any traffic on the street or bicycles have to go through this single road. Yeah, this is the case for cyanobacteria, glaucophytes, and rhodophytes. The green lineage, he duplicated the machinery, lost the phenylalanine residue, and replaced it with serine residues, which are phosphorylated. And you saw that here actually in this slide, you see all these serine residues here on the left for nuclear encoded plastid targeted proteins. Yeah, so that evolved in this lineage, and now you have actually 2 import machineries. So that is now a road with 2 lanes. Yeah, if you want. On the right-hand lane, you can have all the slow folding proteins. So those here on the right that have these beta barrel outer membrane proteins. Yeah, they are slow folding, so it just takes, really in terms of time, it just takes some time for them to be correctly folded and integrated. So every time that here in the red lineage, for instance, a beta barrel protein comes in and gets integrated, no other protein can pass the translocon. Yeah, it just blocks it at that moment. The green lineage has solved that problem by separating the 2 machineries, or the 2 import pathways. Slow folding through the OEP80 and all the normal fast import pathway proteins through the TOX75. And this is also why in the green lineage you have so many additional pathways, for instance, to cope with high light stress and so on and so on. So this is a highly evolved import and very sophisticated, or not highly evolved is the wrong word, but a sophisticated import machinery in a very elegant evolutionary solution that allows these organisms, and this is true for the entire green lineage, to import proteins way more faster, for instance, during high light stress. And this probably also played a role during the origin of land plants that I showed you earlier, because why is this connected? Well, if you think about an algae in water and a land plant, one of the main differences, for instance, is UV stress and light stress and temperature stress and blah, blah, blah. Yeah, so the plastid in land plants needs to adapt way more faster than in an algae. So this was probably a beneficial prerequisite for the conquer of land, that you had these import machineries already established in the green lineage, in the organelle of endosymbiotic origin. And then in the angiosperms, so all flowering plants, for instance, you get one more on top. Yeah, now you have the sp2, which is another duplication event, which is now for protein export. And this protein export channel is probably involved, and I'll go once more back a few slides here. If you think about now the terrestrialization slide again, I showed you on the right-hand side where you have these multiple plastid types. Now, they are also interchangeable, to a certain degree. So you can go from an etioplast to a chloroplast and so on and so on. And this is only, again, the case in the green lineage, and it is probably associated with the additional evolution of this sp2 protein machinery, export machinery. Because some proteins now get actually exported again from the plastid to the cytosol to be degraded and reused for other purposes. So the peptides. Which means that here you have an organelle that can be remodeled. Yeah, so the endosymbiont early on pretty much mimicked the cyanobacterial version, only in an inverted import machinery. And then with time and duplication events, it got more complex and allowed the organelle to actually be way more flexible in terms of protein import and export than its cyanobacterial free-living sister lineages, and all of the cyanobacteria to a certain degree, which is quite fascinating when you think about it. And that takes me to the very last part of today, and time to relax a bit if you want. Usually when I have the students sitting in front of me in class, I ask them, so what do you see here? What does this picture tell you, and what does it all combine? Yeah, and it always takes a few minutes, but usually they get the point. So what do we see? Well, this is a human brain to a certain degree. Here you have the eyeballs, and you've got cats sitting inside the brain, playing around, and here you've got Major Tomcat playing around with two dials that is controlling the brain. Yeah. And at one point, they also get the connection between the cats and the infection by toxoplasma. Yeah, and that's also what the picture is called by the artist Martin Su. And you probably know that there is ideas floating around that what is for certain is that cats are one transmittive agent of the toxoplasma parasite for us. So cats transmit that, we get sick and we get toxoplasma, and the idea is floating around that toxoplasma actually can change the way how we behave. It's been, from what I know, proven in mouse systems, or mice, yeah? So that mice that get infected with toxoplasma, they actually become braver in terms of, searching for food. And that is, of course, for the parasite, again, beneficial, because when the mouse, turns into the brave guy and runs around wildly, it's easier for the cat to catch the mouse, and then for the parasite to hop from the mouse to the cat, because once you have a primary and a secondary host for the parasite. And the same is thought to occur for us humans, so, but of course it's difficult to carry out detailed research on that. But the idea is that a toxoplasma infection in humans actually changes the behavior, especially of males, and so men then become more engaged and braver and whatever. some idea is that they get more attractive, and then the parasite can jump around more easily. But I don't know whether that's been followed up in any more detail. But why Toxoplasma? Well, Toxoplasma belongs to the group of Apicomplexa, and I mentioned them earlier that they actually go back to a once free-living red algae. Yeah. So here you have the dinoflagellates. This is a very simplified tree. Dinoflagellates on the left, so the one that we talked about. Then here, this is Chromera and the Colpodellids. This is a group of algae which has been identified some 10 years ago or so. They, here the Chromerids, they still photosynthesize, and they're sister lineage to the Apicomplexa group. And the Apicomplexa, they have all lost photosynthesis, so none of them can photosynthesize anymore, but only the Cryptosporidia have lost the plastid entirely. All of these other organisms, including Toxo, they still have a plastid, and they're very problematic parasites worldwide. The most familiar ones that you've surely heard about, Plasmodium, which causes malaria, or Toxo that I just talked about, but there's a whole long list of parasites of varying sizes and complexity in terms of their biology. They're a big problem in Africa in particular. I do not know about the Brazilian situation, but I'm sure it's the same, because you've got huge areas in cattle farms where sometimes these parasite infections can really wipe out quite a lot of cattle. So it's a very problematic group. But it was once a free-living red algae. So once being a free-living red algae, they still have a plastid, which in this case, because they are the Apicomplexa, is called the apicoplast. Yeah, you can see it here in the electron microscopic picture, here with AP termed, and it's surrounded by 4 membranes, which you by now should have learned is an indicator for secondary endosymbiosis. And we know that they belong to the, although this was once a free-living red algae that is now evolved into the apicoplast. It's not photosynthetic, but it still makes quite important fatty acids and also isopentenyl diphosphates, and these are also the reasons why the apicoplast is still present. And this is also one of the main drug targets, because of course it's quite beneficial to target these, because if you knock out the biology or biochemistry of the apicoplast, also the parasite dies. And this is also the biology that is conserved in most of the apicoplasts. The apicoplast still has a very rudimentary genome left. So that was sequenced, I think, some 20 years ago now. No, longer, 30 years ago. And that was also when the genome became available, again, the final proof that this really was once a free-living algae. And it was also quite interesting because much of this then moved from the zoology department to the botany department to a degree, because now we were working with an algae. Very small, 35 kb, and I mentioned that earlier. So these are amongst the smallest plastid genomes that we know. And in this case, and for Plasmodium, you actually only have like 2 proteins which are encoded. And the rest that is encoded is the machinery that is there to translate these 2 encoded proteins. And they're somehow involved in protein import. This is what is believed. Here on the right-hand side, just to show you how small the apicoplast is, and that molecular work is surely possible, but very hard on Plasmodium. So in red, you would see the cytoskeleton. This is, I believe, microtubules being stained. And here in the top panel, in green, you see the mitochondria. And then here in the bottom panel, in green, would be the apicoplast. So it's a tiny organelle. I mean, the parasite itself is already very small with some, I think, around 10 micrometers or so, even a bit smaller. So really, really, really tiny reduction of the cyanobacterium down to the bare minimum that is required to fulfill these 2 pathways, which are so essential for the organelle. The entire group of Apicomplexa, they belonged, if you then move one step up in the evolutionary tree, they're parts of the alveolates, and they comprise quite a few groups. The dinoflagellates that we already learned a bit about, these weird critters with the condensed chromosomes, and which have these multiple levels of endosymbiosis. And also the ciliates that we learned about, protists feed on protists, very interesting in terms of general biology, but all of them they belong to the group of alveolata. Why alveolata? Well, because under their plasma membrane, they've got these alveolar sacs. Yeah, so really it is a membrane-bound compartment that all of these three have. In apicomplexa, it's also known as the inner membrane complex. Yeah? So if you should read about that, or if you've heard about the inner membrane complex in the apicomplexan parasites, it's evolutionary homologous to the alveoli and alveolar sacs of dinoflagellates and ciliates. And there's also protein families that then, evolutionary connects all of these three. And surprise, surprise, they're called the alveolins. Yeah, this is one of the families, there are multiple, but this one here actually sits, that's a protein that, if we go back to this slide, stabilizes the alveolar sacs. Yeah? So it's a cytoskeletal protein, intermediate filament protein, that basically stabilizes the cells from underneath the plasma membrane and the alveolar sacs. And what you see here is that the proteins, they vary in length and sizes across the different species, dinoflagellates, apicomplexans, and ciliates, but they have a very conserved motif against which we made an antibody. And then if you stain it, up here you would have DAPI, and you can see very beautifully, actually, in dinoflagellates, these condensed chromosomes that I mentioned earlier. So these massive genomes that are and the permanently condensed chromosomes, and that's why they're so crystal sharp. And then here the Plasmodium parasite, and here the ciliate, always the DNA stained blue. And then you have the alveolin protein, which wraps around just underneath the plasma membrane and the alveolar sacs. And it's one antibody that works across all three groups, dinoflagellates, ciliates, and apicomplexans. And it just demonstrates the evolutionary connection on a molecular level, if you want. And what I always like about this picture as well is if you consider the scale bar. So the scale bar is always identical. These are all single-celled eukaryotes. They all belong to the group of alveolata. But look at here, this little plasmodium cell, which kills millions of us annually, in comparison to this harmless paramecium critter that swims around in our ponds. It would fit in here 100 times or so. So same scale bar, different sizes, different proteins, same molecular function, but which evolutionary unites them, so the alveolates. Malaria, just to wrap today up, was once a free-living red algae, goes back to secondary endosymbiosis of a red algae, and has amazing biology. It's of course a problem for anyone that catches malaria. I actually had malaria. I caught it in India at one point. It's not fun to have. But the biology behind it is amazing. And you probably know its life cycle, quite well studied. It's actually harmless for the mosquito, which is its primary host. So we humans only serve this parasite, this former red algae, secondary red algae, as an amplification tool. When it infects us after the mosquito bite, it first goes into a liver stage, then crawls actually to our blood vessels, and then infects the red blood cells. This is also where most of the research happens, because it's the easiest to culture to a degree, and that's where the parasite just multiplies and multiplies and multiplies in this red blood cell stage, to then at the end of the day be released, be taken up again with the next mosquito bite. And then what happens in the mosquitoes is actually a sexual cycle. So that's where the parasite has all the fun and recombines to then migrate to the salivary glands of the mosquito to close the cycle. And all of this happens with only 9,000 genes roughly encoded in the nucleus. So the genome of the parasite has been reduced, but it hasn't really reduced the morphological complexity or the cell cycle of this parasite. And Plasmodium has had quite an impact on human history. So first of all, the word malaria its stem origin is interesting. It goes back to the Latin for mala aria, which basically means bad air. And sounding simple, but if you think about the time when they coined this term, it was already quite ahead in terms of medical, identifying the source of the problem, because they knew it wasn't the drinking water, they knew it wasn't the food that they were eating, they knew there was something in the air that was distributing this disease. Yeah, so it was bad air quality. Of course, they didn't know that it was a mosquito flying around in the air that was distributing it. We have a minimum of 1.2 million deaths over the last couple of years, at least from what I know. So really a big problem, and of course, unfortunately, in those countries that can't protect themselves against it. It was once, for instance, also circulating around Italy, but no longer is, because they've dried all the ponds. It has a massive impact on the economy, as we see later, and one of the big problems in terms of treating it is that we have multi-resistant strains. It's also believed, and that's what you see here at the bottom. Again, this would be, you know, an interactive picture that with the students I would ask, so what do we see here? And at one point, they would then come to the conclusion, oh yeah, it's actually a baby that we see here, because you can see that the cranum isn't closed. Yeah, so this is just a newborn at the end of the day. So it died a very early and unfortunate death. And this is the way how the Romans then buried babies. Yeah, in this, I don't know the English word, I'm sorry, but in this urn to a degree. And when they then analyzed these, and at around, I think this was 300 to 400, 1,600 years ago, they found many of these, and they found that many of them were actually infested with malaria. Yeah, so there is some historians believe that there's a connection with malaria infection rising and the final fall of the Roman Empire. I mean, there were many reasons, of course, but this was one of them. But not only for the Romans was it problematic. Here on the right-hand side, the guys that you see here, they were all believed to have died from a malaria infection. Yeah, Emperor Titus, Alexander the Great. I don't know who of you have seen the movie Alexander, where Colin Farrell plays Alexander, but at the very end of the movie, you see him laying on the bed, and he's, I think, being offered chicken soup as a remedy. But you see this big fan, and he's sweating like mad, and it's believed that also Alexander died by catching malaria. Actually, when he tried to conquer India. So that's also in the movie. Yeah, he had conquered much of the known world at that time, but he wanted more, migrated east, got to India, and nature struck down on him by infecting him with Plasmodium. And you can also see that here on the left, you've got many guys and emperors, you know, that tried to conquer certain areas of the world that their biology wasn't familiar with. The same is true for Genghis Khan. And then here on the right, you have more poets and artists. Yeah, and there was a time like Caravaggio when, you know, traveling to exotic places became common. And unfortunately, they not only came up with exotic poems, but also exotic diseases, and then ceased to live because they were infected with Plasmodium. So it's believed that, especially very early on during, you know, human history, Plasmodium had a big impact. And again, remember, this was once a free-living red algae. As I mentioned, big problem in terms of the economic impact it has on the countries that are infected. These two pictures, they are a bit blurry, and you'll see later why that is the case. At the top, this is the area where malaria actually strikes quite severely. Yeah? It's not the youngest map, but it roughly works. So around the tropic, or between the Tropic of Cancer and Capricorn, as you all know, that's where malaria is most common. And at the bottom, you see the distribution of GDP, yeah? So economic power, if you want. And only if you then overlay these two maps, and I'll do that now manually live, is when you roughly get an even distribution of the colors. Yeah? And that is because malaria, at the end of the day, its average loss for the economies in these countries is 1.3%. You might think, well, 1.3% is not that much. You know, if I have $100 or $98.7, what's the big difference? Well, this is the average growth rate of the European economy at the moment. Yeah, so the wealth that Europe is increasing, is being lost by these countries here that are struck by Plasmodium. So it is quite severe, and also a number that's you really need to digest. 40% of all health costs in these countries, in particular in Central Africa, they all go to this single disease. There is nothing else like it in Europe or North America, Australia, China, and so on and so on. Yeah, if you think about heart attacks, cancer, and so on and so on, none of these individual diseases cost as much money to deal with as malaria alone. And of course, these countries, they don't only have malaria, they also have cancer and all of the other diseases we have in the Western world to a certain degree. So just this one single critter of secondary red algal origin, endosymbiotic origin, has such a big impact on these nations. So yeah, always keep that in mind when you look at these relationships. Quinine is an interesting story. It's believed to have been used for thousands of years to cure, or not necessarily cure, but at least cope with malaria. And it was, yeah, as I just mentioned, probably used for thousands of years. We don't exactly know, but now once it was synthesized, it was used very early on, because it's known to target the exact biochemistry behind this is actually not fully resolved up to this day, but it somehow interferes with the hemozoin packaging. So what's hemozoin packaging? Well, you saw this in a different format a few slides ago. These gametocytes, they find their way at the end of the day into our blood flow system, yeah, and then they infect red blood cells. And they are, of course, full of hemoglobin, and that's also what the parasite feeds on. But then at the end of the day, it cannot degrade the heme, and it has to be stored. And the way that it does this is by making these semi-crystalline hemozoin packages, yeah. And it's thought that quinine interacts with hemozoin packaging, and by that way then kills the parasite in the blood stage. And because this is biology that is somehow also believed to be linked to the apicoplast and mitochondrion, because you see here this question mark cycling, because they still synthesize it. Yeah, so the balance of heme inside the parasite is interrupted by quinine. Yeah, so somehow these two organelles of endosymbiotic origin come into play when we think about one of these earliest drugs. Today, this is actually not used anymore. Quinine isn't very effective because most of the Plasmodium strains floating around are resistant to quinine. But quinine gave us one nice thing, and that is, I have to do it this way, is gin tonic. Yeah, so British soldiers, think about what I just mentioned with Alexander the Great or Genghis Khan. Yeah, they all want to conquer new territories. The British were just as nuts, and they also went and colonized India, and they had the same problems. Yeah, they had hundreds and thousands of soldiers and British personnel suddenly being affected by malaria. And one of the things that they did is was treat them with quinine. But quinine, which is also the component of tonic, yeah, that is in the gin tonic, so the tonic itself, this gives this very bitter taste to tonic water. Yeah, that's basically quinine that you taste. And because of that, the soldiers were actually allowed back then to mix it with gin. And they actually had established shipping lines from the UK to India, which were transporting gin for the soldiers to mix it. Yeah, and they liked it that much that the returning soldiers basically established it as a drink in the UK. And you all know Queen Mum that died a few years ago, and she got 100 and something. And she always said that the only reason that she got so old is because she had a gin tonic every day. Yeah, but don't try to use gin tonic to cure your malaria infection, because of the resistance that malaria has developed, you would need to drink between 30 and 100 gin tonics every day to treat yourself with malaria. So I think the malaria infection will be the least of your problems if you would try to cure it with gin tonics these days. And as I said, quinine resistance has spread, and this is also biologically quite interesting in terms of its origin and how quickly it spread. Yeah. So here we know that it happened twice independently, 1995 actually in Brazil, and 1957 somewhere in Central Asia, 2 independent point mutations which led to quinine resistance. And back then, traveling wasn't that much a thing. Yeah, so flights were still expensive. People weren't traveling that much. So it took like 2 decades to spread. But today, hardly any strain is not quinine resistant, from what I know. But I'm not really an expert on that. You need to ask the real guys. But what I do know is that new resistances that evolve today, they spread much, much faster because of our traveling behavior. And I think we all, over the last 2 years, experienced how quickly Corona conquered the globe because of our traveling behavior. Yeah. But what's biologically interesting about this is, always keep in mind there was one parasite that had a mutation in a single nucleotide, from what we know, and that was so successful that this single parasite overtook the rest of the population. And that's an evolutionary bottleneck from which this lineage profited. Yeah, so it didn't just cut down the whole population; it cut it down to this one very successful mutation, and it took over the entire population. I think that's quite fascinating, always, when you think about it. I always like to finish with good news, and the good news is that the World Health Organization has just released the first vaccine, for children at risk. So this is a news bulletin from October. Early October this year, so this month at the end of the day. And the last time that I gave this lecture, which was last year, I still told the students, look, yes, they are trying to develop it, but many billions of dollars and decades of research have flown into malaria, and the best recommendation till last year was the mosquito net. So it was still the cheapest and best way to protect yourself against malaria infection was $15 for a mosquito net. This is a game changer, and it's good news for all those countries that are so heavily infected or affected by the malaria infection. And so there's always hope, and hope always gets born with young people doing research. So don't stop, feel motivated. With that, I'd like to thank you guys for your attention, and all the people that have crossed my path and that are part of my former team and the new team. I'm currently floating around between 2 universities, the one in Düsseldorf and one in Marburg. Many partners from around the globe and some funding agencies. And with that, I guess I'm here for some questions as well. Thanks.
Thank you so much. Well, it was a very interesting lecture. And people, if you have questions, we can put in the chat, please. We have some questions here. I can put here in the screen for you.
This is, but that has been proven to be incorrect. So the spirochete type bacterium and the origin of the flagella, this is the idea by Lynn Margulis that I mentioned, and she actually defended that up to her last lecture, I believe. I saw one of her last lectures in Berlin at an endosymbiosis meeting in, when was it? 2014, 15, when did she died a few years ago, and she had the idea that the eukaryotic flagellum also went back to an endosymbiotic event. And they had data back then where they found some DNA at the basal body of the flagellum, which turned out to be an artifact. And we do know today that it was part of this endosymbiotic universe at that time, where it was really thought a bit, and Lynn, who we owe a lot to, was unlimited in the amount of endosymbiotic events that she stitched together to get to the eukaryotic cell. And the spirochete was one of them for the flagellum, but it's turned out to be wrong. So the eukaryotic flagellum originated from different sources. The current idea is actually that very on in eukaryotic evolution, if you think about how our cells divide, chromosome division, so you have closed mitosis, you have the nuclear pore, you have the DNA, you have the microtubular grabbing it, and then they pull apart the chromosomes. Nuclear pore remains, is closed. And we do know that some proteins of the flagella pocket are identical to the nuclear pore complex in eukaryotes. Same proteins. Yeah, many are also homologous, so there is some connection between the flagellar pocket and the nuclear pore complex and the microtubular going through it. And it's thought, and we have a movie out on that, and you could watch it for the origin of the eukaryotic cell. And at the very end, we also implemented that something went wrong with nuclear division, and it ended up in the plasma membrane and generated the eukaryotic flagellum.
Great. I think that Margulies was in Berlin in 11. No, because I watched some lecture of her in Berlin talking about this bacteria.
Yes, it was. Yeah, now that you say it, I was in Berlin. Yeah.
Yeah, great. Well, we have another question,
this is maybe just one more sentence. It really is, especially for the young in the room. It's a nice example of a scientist that has contributed fantastically, to the field, but at the end of her years, just didn't want to listen to the new data anymore.
Yeah, great. Well, we have another question here, but I will introduce with another question before. There are a consensus about the nature of the entrance of the endosymbiont. That is, what is the process? What was this process? Was phagotrophy or invasion, or some invasion by the endosymbiont? And the question, the other question is, what are the main endosymbiont challenges within the phagosome if the phagotrophy is the mode that the endosymbiont enter in the cell?
Okay, so I think the short answer is we really don't know. That is these two models that I introduced at the beginning, right? Where they're competing. We do know that it had to come in somehow. The problem that I see with if it was a phagocytotic mechanism is that you really already would have needed almost a finished eukaryotic cell. Because if you think about phagocytosis and what happens downstream, it is more than just engulfing prey and then it's done. It needs to be degraded, the vacuole needs to be acidified, you need to deal with the products that you then degrade, and so on and so on. That's one of the issues that I see. The other thing being, if prokaryotes can evolve phagocytosis, and they do it, and there is evidence. I mean, there is a new lineage which has a phagocytosis-like mechanism. Paper published 2 or 3 years ago, really fascinating. So yes, there is some evidence that that's happening. It could have been phagocytotic-like entry. I'm open for it, but I think it has its caveats, like the other model has its caveats that says, well, it wasn't phagocytosis. Phagocytosis evolved downstream as a consequence of endosymbiosis. And then the mode of entry could have been, for instance, what the new Asgard archaea model also has, this entangling and then the fusion of archaea. Because eukaryotes, if you think about two protists, ciliates, for instance, during their sexual reproduction, they fuse. And then they exchange genetic material, and then, they divide again. Some archaea can do the same. So they can actually fuse to exchange genetic material. And you can envision, because we do know the current pictures definitely suggest that that's the way how it happened, that it was a syntrophy. They were very close, so that the images that we have of Asgard archaea is these two cells that are glued together, that somehow they were trapped. But the data at the moment is open, and that's also why eukaryogenesis itself is so exciting. And yes, if it was an invasion, and how did it escape the phagosome? Also models out on that, and that's where this idea that it was that the mitochondrion was actually more parasite, and invaded the archaeal cell, which also exists. I mean, rickettsia are close relatives, and that's how people get the connection, because rickettsia are infective bacteria. That it then escaped from being degraded. How? I would guess the same way that many endosymbionts do it today. I mean, if you think about Paramecium busaria, that goes into a relationship with chlorella, because at one point there is a communication between the vacuolar membrane or the phagosomal membrane and the endosymbiont. And Paramecium recognizes, oh, this is chlorella, I don't want to digest it, I actually want to leave it, put it under the plasma membrane, and then use it for its photosynthesis. So I think it would be, early on you would need to establish some kind of communication and mutual benefit, which is, in terms of the origin of eukaryotes, there by syntrophy, because already when they were separated, they needed each other.
Great. I remember that you worked also with membranes and hypothesized about the origin of the membranes of the plastids. There are some hypotheses that explain the fusion of the sternal membrane of secondary plastids, for example, with the membrane of the nucleus.
Mm-hmm. Yeah, that's those organisms that have four membranes, and the one that I can show it. I don't have to talk. I can show it here, and this, yeah. So there you see that these membranes are fused. Yeah, and two models compete. One is that it was phagocytosed, and then the phagosomal membrane fused with the nuclear envelope, or the ER membrane, and then it somehow became one. That has a lot of problems because we don't see anything like that happen in nature. First of all, it's never been observed under the microscope. You cannot, fusing your phagosome with the ER is deadly. I mean, every eukaryote tries to avoid that, because you would spill your phagosomal content into the ER. So what the model that we came up with, and it's published, you could look at the images, unfortunately you do not have them here, is that what we have, for those that know how autophagosomes work, it's the ER wraps around mitochondria, for instance, for degradation. Yeah, so happens every second in our cells. Yeah, mitochondria break, they need to be lysed, some remain, some get degraded, and the autophagosome, which is parts of the ER, just wrap around like two snakes, fuse, and then the content is being degraded. That's the idea that we had for the origin of the two outer membranes, because it also explains, I have to jump to another slide. Here, what is it? Here. So if you look at top right, yeah, where it says Sec61 Selma top tick, yeah. So this is the important thing. Selma is an ER associated degradation machinery. So it sits in the second outermost membrane. It's an ER machinery. So you have an ER machinery sitting here in this membrane, which in our model, this membrane, so the second outermost and the outermost membrane, they're both ER membranes. So this compartment here is basically an autophagosome that was degrading the endosymbiont, fused, and then you get this arrangement. But it's really difficult to explain without the images. But there's also a compartment rule, and that is, it was formulated a long time ago, and that you always have to separate DNA from DNA and DNA from cytosol. So in German it's Plasma from Nicht-Plasma, plasma from non-plasma, by two membranes. And that's why plastids have two membranes, mitochondria have two membranes, the nuclear DNA and nuclear compartment from cytosol, two membranes. And this is different from cytosol to environment, or cytosol to vacuole, and cytosol to ER, which is plasma, because it doesn't have DNA. And that rule, if you apply it to secondary endosymbiosis, you also end up with an easier scenario for two membranes wrapping around and it not being a phagosome origin. But again, I would not sign with my life the one model or the other. Both are plausible and require more research to solve.
People, we have more questions here. Well, I have one more.
I put
Okay.
this is my link. If anyone is curious and wants to watch, I think it's like 5 minutes, 5-minute YouTube video. Of course, I can recommend it because we made it, but it really is. It's so much better at explaining our view on the origin of the eukaryotic cell than I am.
Great.
Yeah. For later, if you
Okay.
haven't already had it.
But it's great. My last question is, why only one lineage of prokaryote became endosymbiont? And complemented, how can be affected all our hypotheses about this origin and the genomes, for example, of Rhodelphs and Picozoa?
Oh, Picozoa. I don't know enough about that. Why only one? You mean eukaryotic origin or plastid?
Okay, no, plastid origin.
Plastid
origin. Yeah.
I mean, we have another case where it happened, and that's Polynella, right? Polynella chromatophora. So there are at least 2 origins of cyanobacteria evolving into a plastid, which still isn't a lot. If you consider that, it's everyday biology of a protist is feeding on cyanobacteria out there in the wild. I think it's really hard because the biochemistry of photosynthesis is a nuclear reactor. Even for a land plant, it's something that, if you think about the mRNA that gets translated in a plant cell, 20%, I think, is dedicated to repairing photosystem 2 and 1 constantly. It's just because it constantly breaks, it constantly is generating reactive oxygen species. And there is hundreds of proteins which communicate between plastid and nucleus, the retrograde signaling, this constant back and forth, controlling the status. It is just biochemistry that's very difficult to regulate. It's great when you have it because you become independent of carbon at the end of the day and food source because you can just make it yourself, but it's very difficult to implement. And the second case with Polynella, which is evolutionary much younger, demonstrates this quite nicely because there you only have a few genes which have already been transferred and get re-imported. And almost all of them have or are associated with light regulation. So now you would think it's random. Evolution is random. There's just all the genes get transferred all the time. But it's especially these that have to do with light regulation, photosynthesis regulation, highlight, stress, changes in lights. Because once photosynthesis is on, you can't switch it off. If the light comes, and this is true for most organisms, light comes, photosystem kicks in, and then the chemistry kicks in, reactive oxygen species get made, and so on and so on. You have photorespiration at night with the mitochondrion. So it's a very problematic cycle, and Polynella, as a younger case, demonstrates that so nicely. They actually struggle with light. I mean, they are phototrophic eukaryotes, but they die, I think, at more than 30 microeinstein. So super low light. They want this cyanobacterium as a plastid, but it's causing a lot of problems. So, and I think that's why it is rare. And the origin of eukaryotes, as that happening only once, the fact that it happened only once should tell us that we should not be looking for, the obvious answer on how it got established. Which is one of the reasons why I think if prokaryotes could evolve complexity by themselves, it would not be a singularity. I think it really is connected with the mitochondria. And maybe directly to your question, if you then look at secondary endosymbiosis, this happened multiple times, right? Because there the eukaryote that was engulfing a eukaryote had all the evolutionary issues already solved by its now endosymbiont, but once host cell that had already dealt with endosymbiosis. So all the light regulation was already controlled by a eukaryotic nuclear system, and it just had to copy paste that. It didn't have to evolve it. And so I think that's why you see secondary endosymbiosis happening so many more times frequently in comparison to primary.
Yeah, then the primary endosymbiosis was a more difficult process than the secondary, for example.
Yes. Because you had pro and eukaryotic system coming together in comparison to eukaryotic.
Oh, great. Well, Professor, we have some compliments in the chat, and I would like to thank you so much. It was very, very, very well, great. Your lecture here, and we learned a lot about this question, and I think that your articles, your papers, will also help us to understand a little bit about the process. Then your citations was very good, but because we will looking for your papers. Thank you so much. Thank you so much.
I enjoy if you enjoy them.
Well, yeah, I custom to say, if you want to tell something about your words, some words, because I always say the last words, but the last words sounds like death. Well, I will kill you, say the last word.
My last words would basically the way how I finished the lecture. Progress and hope for the future is only there if the young get engaged and do the right thing. I think that's really important. Great. Here, the signs of the times. It's only research can save us.
Yeah. Thank you so much, Dan. And até mais, pessoal. À tarde. À tarde nós teremos então a participação do Dr. Sina Edel about Tree of Life also.
Okay. Bye bye, guys.
Tchau, pessoal.